Hello, everyone. Welcome back to lesson number 4 in this concept of the mechanisms of evolution. In lesson 4, we're going to be getting to mechanism 4, which is genetic drift. So far, we've hit mechanisms 1 and 2, natural selection and sexual selection, where we definitely saw some trends in specific directions, whether we're selecting for something or against something. Mechanism 3, we looked at gene flow, just the movement of genes and alleles between gene pools, whether you're immigrating into a pool or emigrating out of a pool. Not always trend-based, sometimes a little random. When you think of the water-based metaphor, it's simple, right? It's kind of straightforward. We're just flowing between pools. Genetic drift, definitely a longer title, longer name, and definitely a little more complex. It's usually a little harder. This is one of the harder mechanisms to grasp just because it's so random. And that's going to be the word of the lesson here. It's all about randomness. So at defining genetic drift, we really start to embrace random changes in gene pools. Very random. No trend at all, right? And how do these happen? Random chance events, right? Just something happens to your population. Something happens to your gene pool. Could not be predicted, could not be planned for, could not be escaped. And based on what exactly is happening, we have different names, right? We kind of call there's two main types of genetic drift that we see happening, and we tend to talk about them in terms of effects, right? We call them like an effect that is happening. So the first one that we look at within genetic drift is called the bottleneck effect. We'll get into that one. Just want to introduce the other one first so we see the two. The other possibility within genetic drift we refer to as being the founder effect. And these two effects, right, they're pretty much just two ways to enact these totally random changes on the gene pool. Totally random, just random chances. So let's start with the first one. Let's start with the bottleneck effect. So here we need to really visualize a bottle, right? Think of just like a glass bottle with the bottle neck right at the top. Kind of cylindrical bottle, but the neck gets even thinner, right? Working up to the mouth of the bottle. It gets very thin, very small. So what's happening here? What's the random chance event? What's the random change that's happening? So picture an entire population of some little organism, right? Maybe they're just like beads or something, right? Not organisms, but I have to pretend a little bit here to get this metaphor. Say that they're living inside of the bottle, right? They're all hanging out in there, doing fine, just surviving, hanging out in the bottle. But then one day, totally randomly, for no reason, right? Just due to chance, someone comes along and kicks the bottle over, right? It's like bouncing along, and individual beads are being sprayed out randomly left and right, and some are being stuck inside, and it's just kind of bouncing randomly right down the street. And then the bottle just rolls, it falls down into a sewer drain, and the bottle is gone. Right? It's just gone. It's totally random. Wow, didn't see that one coming, right? Murphy's Law, almost, right? Anything that can happen will happen. So all you're left with on the ground now are maybe just a few random beads here and there, the ones that just happened to get thrown out and land amongst the field, the grass, right, the yard, the lawn. They're just the only ones that are left, totally randomly. They're the ones that just got thrown out of the bottle before the bottle was completely destroyed and obliterated and washed away down to the sewer. So this is the random chance, right? It's very random which ones came out. And now that's our new gene pool. That's all that survived. We just got a few beans or beads, whatever they are, right? They're the only ones that survived. So whatever genes they have, randomly, whatever alleles they have, that's our new gene pool. And we're going to rebuild from there. Maybe some genes and alleles are completely gone. They were just completely washed away into the sewer with the bottle. Randomly, right? So that was this population going through a bottleneck event, right? Like population bottlenecking. And in real life, right, it's not like populations are living inside of bottles. In real life, this is probably some kind of, like we have here, catastrophic event. Like a tsunami, a volcanic eruption, a mudslide, a flood. It's so hard to survive these events. And it's probably random, just where you are when this event starts, if you can escape and survive and continue to reproduce afterwards, or not. So that's the randomness of the bottleneck effect. That's really one option of seeing genetic drift play out, right? The random change due to random chance in a gene pool. It's a bottleneck effect. The other one here is the founder effect. So not as drastic, or dramatic, maybe I should say, right? Not as dramatic. There's no catastrophe happening here, but it can be just as random. So the founder effect, right? We're talking about founders, for lack of a better word, right? We have a population, we have a gene pool. Think of them like being on a mainland, right? Think of a big continent, think of the mainland. We've got a population living on the coast. And, maybe one day, randomly, right, due to random chance, right, for no reason that we can conceive of, right? It just seems totally random. Maybe 2, maybe 3, maybe 4, right? A very small number of random individuals from this population are just going to leave. They're just going to leave their gene pool. And they're going to go somewhere else, a brand new place together with this little tiny group, little random number of individuals. Maybe they're leaving the mainland to go out to this island where no one lives. Maybe they're birds, right? And they're flying in a coastal breeze and they get blown out there. And they're like, it's too far to fly back. There's a few of us here. Let's just stay here. Let's become the founders for this new gene pool. And it was random. We don't know what genes they brought with them, what alleles. They probably don't have the same diversity as the population on the mainland. But here we go. We got the founder effect. We got a new gene pool. Totally random, totally due to random chance. That's the founder effect. So put a disclaimer on this, right? Especially in speaking in terms of the gene pools, right? This last bit. Very commonly, individuals will confuse the founder effect with gene flow. There's one reason why this is not emigration and immigration. Right, there are founder effect, this being genetic drift. The founder effect implies that there was no one on that island. There is no gene pool for them to immigrate into. Right, they are making a brand new one. They are founding it. So that's what separates the founder effect from gene flow. Just a real quick disclaimer there. That's how you tell those two apart. Might also help to look at some practice here. So let's get into the example. So what's our example down here? We're looking at northern elephant seals. Those are the guys up in the north, right? The Arctic Circle. The males are ginormous, right? The females are quite large too, but the males especially have almost like a small trunk off their faces. They kind of almost look like elephants, aptly named by us, right? But a couple hundred years ago, we started hunting them, right? They got a lot of blubber. Had a variety of reasons back then. We think we hunted them, humans, right? Almost hunted them down to about 20 left. Very small population size, right? Very, very small here. And what's going on? We're hunting them, right? It's just humans coming in. We're the catastrophe in this case, right? We just come in, we blow in out of nowhere. They have no protection against us. We start shooting them. We're hunting them, killing them left and right. So they're just decimated, right? They're just brought way, way down. They're not leaving. They're not going anywhere. We went to them and we just destroyed them. Now, if you analyze their DNA as a population, they're almost identical. It's a big problem for them. There's very low genetic diversity in the elephant seal populations. But let's look at the question here, right? That's what's going on. So what effect is going on here, right? Is it a bottleneck event or is it a founder event or effect, right? What's going on and how can we tell? So first, let's think what we're looking for, right? Are any elephant seals randomly leaving and randomly going somewhere else to set up a new gene pool? No, it doesn't seem like it. Is there some kind of random catastrophic event happening to the elephant seals? Yeah, right? Humans are coming in, wiping them out. So that seems to be the bottleneck effect. And how do we know? Well, you know what? Let's just see what kind of answer options we got here. So first we're looking for the bottleneck effect. So that puts us at A. That's a good one to look at. And B. That's a good one to look at. As of now, we can rule out C with the founder effect and D with the founder effect. These seals are not moving around, right? They're not emigrating from where they currently are to go start a new gene pool. So let's take a closer look at A. Bottleneck effect because there are so many bottles polluting the shorelines where the seals live and reproduce. That didn't seem like it back in 1890, right? It seems like it's more due to hunting, based on what we have in the question. So let's look at B. Seal population was decimated to a point of low gene pool diversity. Yeah, okay. Regardless of any factors of fitness, right? So, yeah, totally random. There it is. Nothing to do with fitness. There we go. So, big degree of randomness here. We got 2 effects that are possible. And as we move into the 5th and final mechanism of mutations, we're still going to keep going with this idea of randomness and random change. So we'll be picking up there next time.